So welcome to the ELIXIR Friday Coffee, where we are presenting the tools and services which are provided by the infrastructure. And today we have the pleasure to welcome our colleague Miroslav Hruška from Palacký University. And he'll be talking about, I think it's a tool which is called Decryptor, which is the online system for detecting peptide variants. So Miroslav, the floor is yours.
Okay, thank you very much for the introduction. So I'm working at the Institute of Molecular and Translational Medicine, which is a part of Palacký University, Olomouc, and today I will be speaking about Decryptor. So as the organizer said, like the system is an online tool for detecting of peptide variants, and I will now shortly say something about the decryptor itself, also what we plan actually to the decryptor, but foremost I will start with the introduction of shotgun proteomics for those who are not really acquainted with the field, to get some idea what it is about. So the shotgun proteomics in general, it's a field which deals with large-scale analysis of proteins. So typically you would have say like a cell lysate or any other complex sample of proteins. And generally, like in shotgun proteomics, the idea is to detect the proteins which are actually present in the sample, and typically to quantify them. Yeah, so these are rather routine tasks which the proteomics does, and such samples are typically also analyzed for a variety of biological conditions, and then one does statistical analysis of the protein expression and see whether some proteins are upregulated or downregulated, and then the person can actually start to think what's going on in the biology. So because of how shotgun proteomics works, the core task is actually detection of peptides, as the proteins are not detected directly. Yeah, so on the right side you can see that so the sample contains like proteins. These are like enzymatically cleaved into peptides, and these peptides are gradually introduced using liquid chromatography to a mass spectrometer. And I will shortly show an example, like how does it look like, or what actually mass spectrometer does. Well, I forgot actually said that the modern shotgun proteomics is generally like heavily tied to mass spectrometry, which is the instrument which actually analyzes the data. So to give you an example of what's going on, so this is so-called MS1 spectrum. So for most purposes, you can think it's actually a graph of masses, like on this axis versus the intensity. And when the molecules actually enter or are actually entering the mass spectrometer, so the mass spectrometer measures their masses and measures their intensities. Technically, it's a little bit different because it actually measures masses to charge, but you can rather easily convert from the mass to charge ratio into mass. So then the mass spectrometer selects a rather like narrow window of these molecules by their mass, and then it subjects them to fragmentation, and what you can see here is actually the masses of fragments. Yeah, like this molecules were isolated, now they are fragmented, and now the core task in shotgun proteomics is actually identify the peptide, which is usually done in such a way that you have either like set of candidate peptides. Here the database can be actually quite large, or there are like many other ways like how you can do it. But what's going on that is that you predict like how the spectrum should look like, and then you evaluate its match. Yeah. So, and once you get the peptides identified, then you detect the proteins, or basically you infer the proteins from the detected peptides. So this was some like rather short introduction to the shotgun proteomics, and now I would say like how Decryptor fits in. So the Decryptor is a system for detecting of peptide variants, and I would note that it's in standard data. So even though like the proteomics experiments typically are interested in proteins and in their expression, and the actual like peptides and their variants are of no routine interest, like Decryptor does exactly this. And you can think of it like it's a protein level analog of variant calling in genetics, because at the end the report will give you a report of variants. So the system is publicly available at this link. So I will shortly present how the Decryptor works, but let me first say something about its architecture so that you have an idea how it works. So what we do is actually we take variants which were globally observed on nucleotide level. These variants are used and introduced to proteins, and these are then like cleaved enzymatically. Like all this happens in silico. This basically happens just once till the database is constructed, and then the database is used in the detection of the variant peptides. So for a researcher in proteomics, like they will just upload the raw MSMS data, and internally, like the Decryptor will do this type of analysis. So which consists like of database searching of the candidate, then it will do some like database independent searches, merges this in analysis, performs like identification of S and V's on the nucleotide level from the observed peptide, and then finally give you a report. So the interface has basically like 4 layers. So you start with the data submission, you get some overall view of the experiment in experiment view, then you can go in for a closer detail in a protein view, and finally you can go to the peptide view. So now I will switch to the online presentation. So let me just change the sharing to another application window. Okay. So I guess you should already see it. If not, please tell me, someone. So I'll go back to the Decryptor website. So the Decryptor resides at this link. You can also take a look at some example, but I will log in. I'm actually logged in, so that won't be a problem. Because the analysis takes some time, I won't really put the MSMS file there, but basically you just select here a particular MS file. Yeah, the file can be rather large. It can be a couple of gigabytes. What you would do is that you will insert the modifications. So it's typically one would use the carbamide methylation, because this is what happens biologically with the experiments. You can put it there, and then you will basically just press decrypt. You will be informed on your mail like when the analysis ends. Well, it's for a file which has, say, this 4 gigabytes, it will probably take around 20 minutes. And so now I will just show you what you will get as a result. So then I'll open the experiment list. So this is just my list of experiments which I was doing, and I will open the last one. And now while it loads, so you get the overall view of the experiment. So for simplicity, I will close this view of the sequence alterations. And in a typical proteomics analysis, you will basically get this lower part, like what proteins are detected, what is their quantity, and based on that, you could then do the analysis of protein expression if you have such a report for multiple biological conditions. But what Decryptor gives you is that you can have these proteins with sequence alterations, and actually it tells you like what alterations to the protein sequences were detected in the analyzed sample. So it also gives you some idea of the quantity. It summarizes the alterations which were found. That there is the detrimental effect which is predicted for these individual alterations. So this can help you to prioritize your like investigations and some source of where these alterations were previously found, or whether there is any disease relevance of the variant. So I'll just open, say, one of these to give an insight to the protein view. So the protein view, again, it's basically a means to more easily get idea like what the protein is about. So you have here like some summarizations, external links if you want to read something about the protein, or whether there is something else available. So that's one thing. Yeah, here you can also see like what's the coverage of the protein, like where these variants actually reside. And for those who are interested more in the individual peptides, one can go through them. Yeah, so this will actually give you mass spectrometry level data, which shows you like what mass to charge ratio of the peptide was measured, like when the retention time sub-measure of confidence, whether there are any other interpretations of the variant, what it could be. And here also it gives you an idea like how much of the sequence was actually reconstructed without the database, and how much of it because in mass spectrometry, like typically you don't really get full support for the whole peptide, but still, for example, in this type, we can see that a lot of this peptide can be like reproduced even without using a database, which gives it some additional idea of the confidence. So so in some sense, like this is what Decryptor does. When you will then when you can also like export the results. So like here you can export the results. Like I will just I can do so. Yeah, you will get the comma separated file to which collects all the variants all together. If you then like take such variants from multiple samples, you can perhaps do some biological analysis what might be going on, and so on. So with this, I would again switch back to the presentation. So give me a second. I will just stop sharing and change it back to the application window of the presentation. Okay. And now I would introduce Claire. So what we plan to do actually with Decryptor is to replace it with Claire. Claire does the very same thing as Decryptor does, but it does in like kind of radically different way, and well, perhaps not radically different, but quite a different way. And I will just say something more about Claire now. So Claire is a next generation of the peptide variant detection. Yeah, and the fundamental like thing which is different is that it exploits differences in peptide prior probabilities. So in proteomics data, what happens is that the molecules actually range over many orders of magnitude, and furthermore, the variants increase the magnitude over which this molecule over which these molecules actually are present in the sample. So to think about the peptide prior probability, you can think of it, it's a probability that a randomly selected molecule from the sample is the molecule of interest. So for example, a reference peptide, which it's kind of shared among the whole population, that one is much more likely than a variant peptide, which is, say, present in a 1,000th of the population. Yeah, so and Claire actually exploits these differences because these differences are present to a very high degree in the analysis of proteomics data. So the system, if someone would like to read more, one can read more about the recent article which we have here about the prior probabilities of peptides. And our aim is to actually replace Decryptor with Claire, as I've said, and the system is in private beta testing as of now. anyway, I would show something about it. So to give you an idea what the architecture of the Claire is about, it's in some sense, it's very similar. Yeah, it again starts with the database. It performs like then the variant search. But the different thing is here when we do like deep analysis based on the prior probabilities of peptides. So the database for such analysis has around like 0.5 terabyte. So it really tests a lot of possibilities, like what the peptide could be, and really tries to derive the peptide with high confidence. And I would just note that this is especially important because like some somatic variants are, say, on the order of 10 to minus 6 less likely than a reference peptide, and this actually changes the situation quite dramatically. Yeah, so for example, if some criterion for a peptide to be correct works very well for reference peptide, it can be like absolutely inadequate if you want to identify peptide which is like much less likely a priori. Yeah. So now I will just show something which you can do with Claire. Possibly you could do something similar with Decryptor, but given that I will speak about both, I will now focus on Claire. So one thing what one can do is to detect the hypermutation status of a tumor. So the hypermutation status is a clinically relevant parameter because it actually can help to select the therapy. So, for example, it can indicate that the patient will likely respond to immunotherapeutic treatment of cancer. So here we showed on the analysis of NCI60 panel, which contains like 59 cell lines, that if we compare like detected germline and somatic variants, so those samples which have deficiencies in DNA repair mechanisms have actually like much larger rates of protein mutations, and we actually saw the same when we did analysis of patient samples. So, for example, here, like on this left side, you see that the protein mutation rates in those which didn't have deficiencies in the DNA repair mechanisms were like much lower than those which had the deficiencies. For comparison, we also included the analysis on the level of DNA. So, of course, the differences are like much more like clear cut. Just for control, we showed that like this doesn't happen to germline variants. There are no differences, and perhaps one like biological interesting like situation is that we correlated the protein mutation rates obtained on the DNA and protein level. And although there is like some correspondence, there are also situations when the rate of mutations on the level of DNA was like rather low, while the rate of the mutation on the protein was rather high. And so it would be, of course, interesting to see whether the patients would actually respond better based on the protein mutation rate or DNA mutation rates. So another thing which one can do is actually like to indicate the tumor stage from the protein mutation rate, although it's by far not really clear cut that one can do that because the number of variants is typically low, and especially in patient samples. But it showed some promise in the sense that the protein mutation rates like grew over the stages, similarly as they did for the DNA mutants. Here again as a control that nothing is really going on when you are interested in germline variants. One can also use this Declare to identify individuals from proteins against DNA database. So we have here a data set of samples which consist of 7 family members. One of those are monozygotic twin sons, and actually we can calculate the probability that the DNA sample, or actually that the protein sample corresponds to the DNA sample. So this we also use to identify like cell lines using peptide variants, and I will show something soon in the interface for Claire. So let me again switch to the browser. Okay, share. And so in some sense, like the Claire is similar. So the interface is kind of similar, but I will just give you an idea like how the interface looks like. So here we have quite a few samples analyzed. Yeah, some of these are like very deep samples. So this one, for example, contains more than like 600 identified variants. This is rather rare. The sample is not really a typical proteomic sample, but anyway, it can give you an idea that the number of variants detected can be actually quite high. Like the Claire also does automatically that it authenticates the origin of cell line. So for example, we observed that in some of like public NCI 60 data, so there are some discrepancies. So for example, I'm not sure whether I'll find it here now, but we actually realized that what claims to be a particular cell line is actually another one. One can also take a look like how does it look with other tentative origins. But for example, this one is identified with a really high probability. Yeah. So similarly, one can then go through the individual detected peptides. As it was in Decryptor, the interface is in some sense expanded in the sense like what it can give you. Again, yeah, like basically the same. There is the list of variants, whether they are somatic, germline, yeah, like the protein abundance. So I would just select, say, for example, this one. So again, you have the protein view, detected proteins, peptides, like some prediction of its harm by like a variety of predictors, some details about the alterations, where it was found, in what type of databases. So if I'll click here, this will give me like a closer look over the peptide view, similarly as Decryptor did. But Claire also allows you to do like a very, very detailed view of the peptides. Which also shows like the results of the deep search. Like the deep search considers for somatic variants more than few millions of candidates, like for a given mass range. So you can also have some idea like how really what is the next best interpretation of the spectrum. The analyses are like really, really quite deep in this way. And similarly as with the Decryptor, you can then export the results, and if you do this for multiple biological samples, then you can start to create some analysis similarly as we did with the mutation rates and so on. Okay, so with this I would again like switch to the presentation. Okay, and so I will basically now conclude. So Decryptor is a publicly available system, as it was seen for detection of peptide variants. I would note that it's mostly usable for protein polymorphisms. The situation with the somatic variants is like more complicated, as I've said, that their prior probabilities are like much lower, and actually to detect them at the same level of confidence, you need to perform really enormous amount of effort in terms of like computations. So we aim to replace Decryptor by Claire, and the system is capable of detecting reliably a wider class of variants. And basically, as you have seen, the Claire has potential applications in multiple fields, including cancer research, forensics, and the research reproducibility. So yeah, that will be basically all. I would also like to acknowledge the ELIXIR infrastructure. So thank you. That's
Okay,
all on my side.
Thank you very much for nice introduction to the tool. We have a few questions here, and the first question is the Decryptor publicly available or upon request, and how would you compare it with others available tools?
So the Decryptor, Decryptor is publicly available. So it's like there is no need for request. You just basically register, and you just confirm your email, and you can start to do your analysis. Comparing it to the others, well, that's a good question. So the Decryptor was made like a couple of years ago at times when there wasn't really that many competition in this field. Now I am aware of systems which are like standalone system, like for example tag graph or some others which can do analysis of this kind. I am not aware of a tool which does this online. So yeah, there are certainly options like what one can do to detect peptide variants with a standalone system, and I'm not aware about one which does it online. So that will be the answer.
Okay. Carol asking, where do you get info about sequence alteration effects?
It's actually from DB-NSFP database. It's a database which compiles data from, at that time it was 20 predictors. So it's very useful. It's done on ensemble reference genome. Yeah, like because we also work with the ensemble proteins. So for us, it was very straightforward to do the correspondence. So it's all pre-computed. I know the database is a couple of gigabytes, but then you just put there the variant and it will just give you all the results from all the predictors.
Okay, let me ask you the last, probably heretic question. So how reliable are the outputs of your Decryptor? And did you ever try to compare it with the results on some other tools?
Yeah. So the results on the Decryptor are reliable as long as you are interested in the rather protein polymorphisms. So they are not that reliable with the somatic variants, but I would just know that with the somatic variants, it's actually quite complicated to even validate the detection of somatic variants because like they also don't have to really be in the DNA or mRNA sample. And sometimes it's really not straightforward to tell like what is actually true and what is actually false. With Decryptor, we did compare it, for example, with some exhaustive searches using Xtandem. Like if one doesn't perform additional analysis of the data, like it will be like a lot of stuff will be incorrect. So one thing what one can really do is that you can use basically any software which detects peptide variants, but you should be aware whether the peptide variants detected are somatic or germline variants, and ideally you should also know their population frequency, because those who have like high population frequency are actually quite likely a priori, so these will be very likely correct. But those who are priori unlikely, which might be some rare somatic variants, they would be for the very same settings of criteria very likely incorrect. So it's a kind of puzzling situation. And then what Declare does is that it actually incorporates the prior probabilities into account and it deals with them accordingly. Yeah.
Okay, thank you. So because there is no other question, Miroslav, thank you very much again. And
thank you.
For others, see you next Friday on the ELIXIR Friday Coffee. Bye-bye and have a nice day.
Thank you. Thank you. Bye-bye.